So this diagram depicts a nice cartoon representation of how genes are introduced into a target cell, yes, into the nucleus. So please note that the transgene is a gene which is being transferred, yes, the organism which receives it. If it is done at the embryonic level, the gene is present throughout the genome of the organism. That is the reason why this organism becomes transgenic. So students, please remember that there are different ways of introducing DNA cells or nucleic acids. So one of the major methods is the transformation. That is how you introduce your plasmid into the target bacteria. Yes, that is transformation. So that is a calcium chloride mediated methods. All of you know that calcium chloride is used to, or to say, make sure that the cell becomes competent enough. So what is competent cell? Competent cell is that cell which will be able to take up your plasmid DNA. Yes, and undergo transformation. So why calcium chloride only? That reason will be, what to say, known in today's PowerPoint. Yes, we will come to why only calcium chloride is being used. Okay, and that is this method of transformation. That is this method of what to say, transformation. Next, transduction. You know what is transduction? Transduction is bacteriophages in order to deliver your target DNA into the bacterial cells. But it's not just that. Viral delivery, what to say, viruses can be used to deliver your gene of interest even into animal cells or even plant cells. So that is why we have this chapter called viral, what to say, vectors, which perform delivery of your target DNA using specific viruses like adenovirus or retroviruses or virus-like particles. Okay, so you have studied about how this happens in bacteria, right? In the chapter on what to say, phagements and cosmets. So how do you use Fajmet? It is a blue script, M13 blue script. Can anybody tell me how it is being used? It has to be used along with a helper phage. Otherwise, you will not be able to achieve proper infection and replication of your Fajmet DNA within your target bacterial cell. Yes, all of you know about this. So therefore, it is very much essential, even in this case, that you are using viruses in order to, what is it, transfer your target gene to the your animal cell, or even in a plant cell also, you need to use a helper phage. The helper phage makes sure that this particular viral particle, which has your target gene, it successfully gets infected into your target cell, but it will not replicate. It will get successfully integrated into the genome. Okay, the only difference between plasmids and this viral, what to say, uh, transfer strategies is that in bacteriophage, your purpose was to replicate your DNA. Yes, the virions had to replicate within your bacteria, and you have to successfully get enough number of copies of your plasmid. But here, the purpose is to, what to say, permanently integrate your gene of interest into the target organism. So therefore, we have three different methods. One is the transfection methods. You use different methods to transfer your gene of interest into the eukaryotic organism. So transfection is usually referred to eukaryotic organisms, whereas transformation is referred to prokaryotic organisms, like introducing a gene of interest into bacteria. Okay. So all of you understood the terminologies: transfection, transformation, and transfection. So you could be asked a 2 or 3 mark question. Mention the terminologies which explain the transfer of nucleic acids into target cells. So which are the 3 mechanisms? Transfection, transformation, and transduction or viral infection. Yes, this could be asked as a 3 mark question. Please remember this. Okay, so this is transformation, how it is introduced into bacterial cells. Transfection, that is the topic of today's lecture, and transformation, how in what to say, I'm sorry, this is supposed to be, yeah, transformation of cancerous cells. So using a viral particle, you can immortalize even cells. So there are 2 different kinds of animal cells which can divide continuously, or which are continuous cell lines. You know the difference between primary and continuous cell lines, right? In animal cells, primary cells are those which are derived from a specific tissue, like from epithelial tissue, or from, what to say, from lymphocytes, or they could be from, what to say, liver cells, like hepatocytes. Yes, it could be neurons, different kidney cells, different ones. But these cells, when removed out of the body, they will be able to divide only for a few generations, after which their division completely stops. What if you have to achieve continuous dividing cell lines? For that purpose, what do you use? Either you make them cancerous, either they have to be cancerous cells, hepatoma cells, yes, lymphoma. If you have a cancer cell line, it will have the innate capacity to divide for a long time. Whereas if you have a primary cell line, and if you have to, what to say, induce it to undergo several divisions, that is what is called as immortalization of a given cell line. So what do? You will remove some of the inhibitory factors for the cell, which will prevent it from dividing. In the cell cycle, there are a lot of checkpoints, after which, if the checkpoints are active, they will not allow a cell to divide. Similarly, these primary cells also, after, what to say, a certain amount of time, they will stop their cell division. If you are able to remove these obstacles in their cell cycle, what to say, checkpoints, to allow them to divide indefinitely, that is called as immortalization of these animal cell lines. That is what is being given over here. So a given eukaryotic cell, if it is sometimes infected by a virus, you introduce certain genes, you immortalize that cell line. There's a difference between immortalized cell line and a cancerous cell line. Cancerous cell line, it is being derived out of an actual cancerous tissue, whereas immortalized cell line, you take a primary cell and you infect it with the virus so that it will divide indefinitely. That is also using the viral, delivery method. Okay, so which are all the molecules which can be transfected? You can transfect a plasmid, you can transfect RNA, you can transfect even siRNA for inhibition of certain mRNAs. Now, in transfection, you have something called as a transient transfection and the stable transfection. So what is the difference? Temporary and permanent. If the DNA gets integrated into the host genome, it is called as stable transfection. Usually, transgenic organisms are stably transfected organisms. So you will be able to introduce your gene of interest into the genome of the organism. In that case, it becomes a permanent transfection. Whereas if your DNA, it doesn't integrate into the genome and exists in the extra, nuclear space, then that becomes, transient transfection. So transient transfection, usually it will be active for about 24 to 72 hours, sometimes 96 hours, after which sometimes what will happen, the DNA might be degraded, whatever the DNA you have introduced. Okay, so this is used to silence certain gene expressions. Okay, that is the purpose of transient transfection. For stable transfection, usually you look for knockout animals. You delete a gene completely in a given organism by producing a transgenic organism. Or let us say you produce a transgenic organism where the organism is able to produce a particular protein of interest, and maybe it might manifest certain color on its skin surface, or have more amount of hair, or better immune system, such different things. So that is a stable transfection. Now let us look at the differences between stable and transient transfection. So students, this could be out fast for 3 or 4 months for comparing transient and stable transfection. Please remember, this will be a very important question for your examination point of view. So I could ask this as one of the questions for your open book exam also. Yes. So 1 second. Yes. So transient transfection. In transient transfection, what happens is that the transfected DNA is not integrated into the genome. Yes, but it remains within the nucleus. Yes, because it is a eukaryotic cell, it will remain within the nucleus, but it does not integrate into the genomic DNA. Whereas in the case of stable transfection, it is integrated into the genomic DNA. In the case of transient transfection, the transfected material is not passed on to the progeny, so it remains only in the parent cell. Yes, the genetic alteration is not permanent, whereas in stable transfection, once you create, yes, a transgenic organism, if it reproduces, if it is, let us say, a cell line, how many times it divides? Even in the first and the second generation of cells also, you will be able to find your gene of interest. If it is an organism, even in the F1 and F2 generations also, you're going to find your gene of interest. So it does not require any specific selection. Yes. So stable transfection requires specific selection for the isolation of stable transfectants. So when you are doing transfection. It might, the gene of interest might integrate into the genome, or it could stay within the nucleus, as in the case of transient transfection. Okay, so if you have to make sure that your transfected gene integrates into the genome successfully, you need to screen the transfectants. That is very much required in stable transfection. So both DNA as well as RNA can be used for transient transfection, because RNA is unstable after some time. After it expresses after some time, it will be degraded within the host cell. So therefore, for transient transfection, even RNA can be used. Whereas for the stable transfection, compulsorily only DNA, molecules have to be used. So high copy number of the transfected genetic material results in higher level of protein expression. So if you need a very good expression, yes, mostly the transformation, whatever you achieve, that is presence of plasmids within extra chromosomal DNA within your bacterial cell, it will be maintained. But as I told you, we had a very elaborate discussion about this. So what about copy numbers? So for some reason, the plasmids are distributed to daughter cells. Will they maintain their copy number? Yes, copy number, it will definitely maintain. So in the case of bacteria, it does maintain. And beyond a certain level, the plasmid should not replicate. But that is different. Though it is extra chromosomal in the case of bacteria, it is stably maintained within the bacterial cell. Whereas in the case of eukaryotic cells, if you have certain, eukaryotic cells or animal cells, eukaryotic means I can refer to them even as yeast cells, the microbial cells. If you have the animal cells or plant cells which are having extra chromosomal DNA, it sometimes happens that the cell will not support it after some time. Yes, as the cells keep dividing, the plasmid might not be retained. So therefore, copy number might be very high within the first target cell. Okay, but as it keeps dividing, it quite happens that the copy number will go down with number of cell divisions. This is only in the case of this kind of eukaryotic cells. But in bacteria, it will maintain the copy number. So please don't get confused over here. Copy number, this is transfected. Once you mention the word transfected, it is only for eukaryotic cell. If I ask you a multiple choice questions, if transfection is for eukaryotic or prokaryotic cells, you have to mention that transfection is only for eukaryotic cells. You mention the word transformation, then it is for prokaryotic bacterial cells. So as in the case of transfection, transient transfection, sometimes if you need higher levels of protein expression, you can introduce your gene of interest or your RNA of interest in larger quantities. You will have a very high expression of your gene in the first generation. After the cell divides, you will lose your copy number. Whereas in the case of stable transfection, the protein can get expressed only to a certain level as demanded by the host cell machinery. Yes. In the case of transient transfection, cells are typically harvested within 24 to 96 hours of transfection. Okay, because within 96 hours, your entire transfection, your process, it will lose its quality. Okay, so as I told you, as I was telling you over here, yes. So usually the effect on the gene expression in the case of transient transfection is only up to 96 hours. That is up to 4 days. Beyond which you will not be able to see the expression of your target gene. So therefore, it is a temporary phenomenon. That is only for expression of certain proteins. Let us say you have transfected a given animal cell. Yes, using liposuction, you're having one expression vector, and you're going to express a certain protein in that, and you're going to look at the effect only for 4 days. After which it is gone. DNA is no longer maintained, or even if it is maintained, it will not be expressed successfully. Whereas stable transfection, it will take 2 to 3 weeks to isolate and maintain your transgenic organism or transgenic cells. Okay, you can have transgenic cell lines also wherein a given gene could be knocked out. Knocked out means what will happen? Let us say there is one particular gene necessary for infection or producing certain virulence. Okay, your job is to remove that gene. This virulence gene will have 2 flanking regions. You will determine the sequence of this 2 flanking regions, and you will design a plasmid, okay, where you're going to have a blank cassette. Blank cassette means just a small piece of DNA which has no role, or it could have a selectable marker gene such as an antibiotic resistant gene or something like that. So if these 2 flanking sequences are there, so what will happen? The flanking sequences they will undergo recombination, and this gene of interest will be pushed to the plasmid. I'll come to that. Crossing over takes place. So because of this crossing over, what will happen? This blank gene is introduced in place of your virulence gene, and the virulence gene goes to the plasmid. The plasmid is no longer maintained; it is degraded within the post-eukaryotic cell. Yes. So therefore, your gene is now getting interrupted. You no longer your gene is there where the locus is there. It is instead being replaced by a DNA fragment which could have an antibiotic resistance gene. Okay. So that is the reason why it takes about 2 to 3 weeks for this entire process to get over. So transient transfection is usually not suitable for studies using inducible promoters. So if inducible promoters are there, it is not going to be very useful. The reason why, because if inducible promoters are there, they usually act on these inducible promoters at once, which should be part of the genomic DNA. That is the reason why for transient transfection, we don't use inducible promoters. Now, the purpose of transfection, in order to study gene function, it is quite necessary for us to perform the process of transfection. Yes. So let us say you need to produce antibodies or vaccines or hormones by animal cells. All of you know that antibodies can be produced only by the plasma cells. So studied in immunology, the B cell, when it gets activated after being exposed, to cytokines which are being produced by the T helper cells. So the B cells will get activated and differentiated into plasma cells, which produce a lot of antibodies. Let us say you need to engineer your plasma cell to produce a different kind of an antibody. Quite happens, you will be able to do that, but for that, you need to do a good transfection strategy. The vaccines and hormones can also be produced. Usually they're produced by animal cells. Yes, you can study protein expression in eukaryotic cells, how the protein expression takes place. Yes, and you can also do an embryonic transfer. That is, if you can transfer your target gene to the embryonic stem cells, you can produce, as I said, transgenic organs. Yes. Let us say there is a deceased mouse. Yes, the mouse is having a genetic disease. Okay, it is, let us say, not able to produce much of lymphocytes. Okay, some reason you have created, or it is a naturally available mutant. Let us say you have identified a gene, then transfected with this gene, the cancer is able to, get overcome. So you can introduce this gene at the embryonic level and see if this cancer is overcome by this particular gene. Okay. So these are the purposes. One is for studying gene function and regulation, studying protein expression, and in order to produce transgenic organisms. They can do it either at the cell line level, or you can transfer the tissues to the organs within humans. Let us say a person is having some liver disease, okay, and it is a genetic disease. Let us say, so the livers are not able to metabolize certain drugs properly. So what do? You remove the hepatocytes from the liver. Okay, culture them in vitro in the laboratory. So you perform this gene therapy. You know what is gene therapy? Deliver genes directly into the target cells, and the genes will start functioning properly. And because when the gene starts functioning properly, the cell resumes its normal function. That is called as a gene therapy. Instead of giving protein molecules or hormones in order to have therapeutic function, instead you give genes itself into the cells, introduce them into the cells, so that the product of the gene will make the cell function normally. Okay, that is gene therapy. Most of you would have studied that. Okay, fortunately now gene therapy has slowly started to see some light. Okay, it is not that it is only theoretical. No, it takes a lot of time. Yes, gene therapy has slightly come into effect. So what do? You deliver this gene into the target cell within the in vitro conditions and allow the cells to divide for some generations and put back the cell into the organ from which it was taken. So then when the cells start dividing within the in vivo conditions, that is within the organism, it quite happens that the target organ, resumes its normal function. So this is gene therapy. Some cases what people do, they infect a particular patient with a viral based gene therapy, where the virus will deliver the gene of interest into the target cells, and the gene of interest from the virus, it will enter the target cells, and slowly it will get integrated at the genomic level, leading to stable transfection. Now, what are the factors which influence transfection efficiency? The cell type, what kind of cell you are using, okay, from which tissue it has been taken, that is very much determined, that will determine the efficiency of your transfection. The health of the cells, okay, the age of the cell, okay, from where you're taking, that will depend. Confluency, what is confluency? How well the cell density is there in a given area. And serum. So all of you have studied what are the different components of serum, right? What does serum contain, students? What does it contain? It contains different differentiation factors, right? So different differentiation factors which will control and regulate the differentiation of specific cell lines. Will have different proteins for nourishment also. Therefore, serum is a vital aspect for the growth, okay, and development of animal cells and even maintenance of animal cells. So you almost must be knowing that any animal cell culture media, you must have studied this in your first semester in analytical biochemistry 1, because I had taught detail about animal cell culture. So for that, whatever medium, modified medium you use, that should have serum along with it. You have the fetal calf serum, fetal bovine serum. So all these things also determine the transfection efficiency. Why? Because the cells have to function normally in the medium first. Taking a class, let us say, if let us say you're having a lot of tension in your brain, oh, tomorrow exam is there, or let us say you're hungry, oh, I'm very hungry, whatever the restaurant doesn't go to my mind. Instead, if the teacher allows, if I allow you to have food and listen, you keep enjoying your food, enjoying your coffee, and whatever I say, you will keep to understand better. And that's the reason why most of us recommend that when you come for classes, parents might tell you that once you have proper food and come, you'll be able to focus better. That is one of the give you an example. So therefore, even for transfection to happen properly, the environmental conditions of the cells which get transfected are very important, and time also, there should be ample amount of time given. So which takes more time, stable or transient transfection. Stable transfection takes a lot of time as compared to transient transfection. And your DNA quality and quantity also matters a lot. You make sure that whatever DNA you purify, could be your plasmid DNA or your viral based components, the quality of DNA has to be of very good, what to say, good quality, should be pure, should not have any contaminant DNA. That is, how to say, a non-recombinant vector being there, something such thing should not happen, because that will affect the quality of your transcription process, and it should be in quite good amount. The quantity should be good. Now, so what is bacterial vectors? So you can something it might intrigue you, this condition. So why are we speaking about bacterial vectors? Bacterial vectors means bacteria themselves act as temporary hosts in order to transfer your gene of interest into another organism. That is called as bacteria being living hosts. Example is Agrobacterium tumefaciens, which will be done in the next class, coming classes. Agrobacterium tumefaciens, yes, it is a bacterium which will cause crown gall disease in in plants. Okay, so what will happen? The point at which the Agrobacterium infects, the plant tissue will start dividing. The plant cells will start dividing into a mass of cells which is rich in this Agrobacterium, and it will appear as a tumor, a huge, what to say, swollen area within the plant. Okay, so this is what is called as a crown gall disease. So this was identified by the botanists, by the agriculturists. But you will be able to use Agrobacterium as one of the, what to say, host organisms or one of the intermediate organisms to transfer your gene of interest into the plants. How can you do, what to say, introduction of genes into plants? It is very difficult actually to do transfer genes into the plant. You're doing a microinjection method at embryonic stem cell level. You can take the embryonic stem cells, the protoplast, and introduce a gene of interest. Yes, that is for transgenic plants. But the plant has already developed, let us say it has developed certain disease, and you want to render certain resistance against that disease, okay, to the plants. So how are you going to do that? The way to do that is use agrobacterium humifications. It's one of the best used methods. So you introduce first your gene of interest into agrobacterium, then make the agrobacterium infect the host plant. Okay, but there is one thing to be noted over here. The agrobacterium which infects your plants to transfer the gene of interest, it should not cause any disease within the plants. Okay, it should be only able to cause temporary localized, cell division, and make sure that this bacterium is present within the localized mass of cells within the plants and be able to transfer the gene of interest from the agrobacterium to the plants. Okay, so that is what is called as bacteriotransfer, and agrobacterium is one of the agents used for bacteriotransfection or bacterofection, bacterial transfection or bacterofection. So this could be asked as a 2-mark question. What is bacterofection? Bacterofection is a process wherein bacterial cells, living bacterial cells, are used for gene transfer into eukaryotic cells. Okay, where the bacterial cells, they serve as intermediate hosts in order to transfer your gene of interest. Okay, so the bacteria invades the, this could happen even in animals also. This is described for plants. So bacteria invades the host animal cells, undergoes lysis, releasing their plasmid DNA. Examples of these are Salmonella species like Listeria monocytogenes and Shigella flexneri. So lysis of these two species after they escape the vesicle. Okay, so the plasmid DNA then finds its way into the nucleus and gets incorporated into the genomic DNA. Okay, you can mention Salmonella, species is Listeria monocytogenes and Shigella flexneri. So these bacteria, into these you are going to introduce a gene of interest and allow these bacteria to infect the animal cells. So once these bacteria infect the animal cells, you should make sure that they don't cause any disease to the animal cells or the animal tissue. Okay, once they infect the host animal cells, these bacteria are destroyed. They enter into the cells, the bacteria are destroyed, but the gene which is there on the plasmid and within the bacterium, it gets released into the host animal cell, and then it will be able to get integrated into the genomic DNA. This is the entire process of bacterofection. First, you introduce a gene of interest into bacteria and allow that bacteria to infect your host target eukaryotic cell. Okay, all of you understood? This could be asked for a 2-mark question. Okay, now within the transfection, how are you going to plan this entire thing? First, what do? You do the tissue culture, you count your cells, the cells which have to undergo transfection. Resuspend the cells in whatever buffer is not electroporation. I'm just telling you, whichever you can make it as transfection buffer. You add your nucleic acids and you perform your transfection method, whichever method, electroporation, lipoaffection, or gene gun method, whatever it is. So after transfection, next you plate the cells. Okay, then you do an analysis. Take, let us say there are different on the ELISA plate, different 96 wells are there. Okay, they've done the experiment and all the 96 wells they also done the transfection. Aliquot each amount of cell and see, analyze them either by microscopy or looking for gene expression, either protein expression or for gene expression. You will come to know whether your gene of interest has been stably integrated or not. One by microscopy you can do. If it has any GFP tag or something, it will give you some fluorescence. You can see that yes, your gene of interest has been stably incorporated into your cells. Is one of the methods. The other thing is looking for protein expression. Okay, you do a Western blot analysis to see whether your gene of interest has expressed as a protein, or look for the reporter gene activity. If it is for luciferase, this you can have luciferin and luciferase, and if you get some light, luciferin is converted to oxyluciferin. If you get some light, you will come to know that yes, my gene of interest has been integrated stably. Or you can look for gene expression. Yes, you can do real-time PCR. You can do that, or you can do flow cytometry, looking for the surface molecules and see if these molecules are there by having antibodies against them. So this is the overall workflow of transfection. This could be asked for a 3-mark question. Describe the methodology used in transfection, or describe the workflow of methodology used in transfection. The first, you need to select your cells to be transfected. Let's resuspend them in, I'm sorry, I'll remove this later. This is transfection buffer, or the respective buffer used for transfection. You add the nucleic acid, that is your source DNA. It could be a plasmid DNA, or it could be a virus-associated DNA, whatever it is. Add your DNA. The DNA could be coated with any beads or something like that, if it is the case of gene gun method. Then you add it to your cells, which need to be transfected. Then you perform the method of transfection, whatever method has been you have selected. After you do that, you plate the cells. You transfer them to a culture medium. You grow them. After you grow them, then you analyze them. Look whether your gene of interest has been expressed or not. How do you do that? 5 methods. 3 broad methods. One is look for microscopy. Is there any change in morphology of the cells? Second, look for gene expression. You can do RT-PCR to see whether your RT-PCR or real-time PCR to see whether your gene of interest has been there. Okay, you can do even flow cytometry also if your gene is being expressed on the surface of the cell. So if it is being expressed on the surface of the cell, you can use certain antibodies to bind to the surface cell receptors, the antigens, and based on the antibody, what to say, fluorescence, whatever it gives. Okay, you can sort the cells into different compartments. Okay, because the flow cytometer, it will give certain charge to these antibodies, and it will ask it to get segregated. So if you get a good population amount of cells, you can find that yes, so these cells out of 10% of this entire transfected population, it has my gene of interest. Okay, the other one is looking for protein expression by if your gene of interest, if your target gene has any reporter gene, you look for the reporter gene activity. Let us say you have a luciferase gene, which is upstream of your target gene, you will look for luciferase activity, or you do a Western blot to confirm the protein expression. Now we can broadly classify. One second. So we can broadly classify the transfection methods into 3 categories. Okay. So what are these methods? One is chemical-based methods. So use certain chemicals in order to perform your method of transfection. They're usually a cationic lipids. Okay, you can this is also called as lipoaffection. You can use calcium phosphate or calcium chloride. Calcium chloride is what is used for the bacterial transformation. Calcium phosphate is used to kind of prostrate DNA and introduce it into your target cells. You can have different cationic polymers. Okay, cationic polymers are positively charged different polymers which will start interacting stably with your DNA molecule. Okay, and help it to enter the cells. And DEAE dextran, diethyl ammonethyl dextran. So that is also quite cationic in nature. Okay, dextran is a naturally available polymer. Okay, so which is covalently linked to diethyl aminoethyl. So that is too is positively charged because of the amino group which is being present. The amino group will start interacting with your negative charge of your sugar phosphate backbone, and that will help it to enter the cells. Then we have magnet-mediated transfection. This is a very interesting method where you have a magnet-based, what to say, transfer of your gene into your target cell, and you have activated dendrimers. So today we'll study all the chemical, what to say, based methods. Instrument-based, we can study a few of them today. Yes, and rest of them we'll see. If we have time, we'll be able to study. Instrument-based means you need a specific instrument in order to introduce your gene of interest. Electroporation. Add a slight small current. When you add a little bit of current, what will happen in your target cells? There will be different pores formed because of the current. Yes, what will happen in transmembrane proteins? They will undergo certain conformational changes so that it will allow DNA to pass into a cell. Okay, so of course that is a temporary condition. During this condition, when pores are created, it will allow the passage of DNA. That is in the case of electroporation. Microinjection, you use a microinjection needle. Usually this is used for embryonic transfer. This microinjection needle is used to pierce into your target cell and transfer your DNA of interest, your gene of interest. Okay. Biologics particle delivery is nothing but the gene gun method. You coat your DNA onto certain beads, gold beads or any other metal beads, and use a gun to fire it into the target tissue. It seems kind of very amusing or something strange. So if you have the target tissue, it is fired inside. Okay, because of the projectile movement, the momentum, what will happen? The momentum is actually created by your particle. It could be a metal particle onto which your DNA is coated, platinum or gold particles. Okay, onto which your DNA is coated. You force this to forcefully enter your target cell. Because of this momentum, your target gene is able to enter your eukaryotic cell. That is called as biologic particle delivery or the gene conversion. Yes, the landmark is called as a laser friction or opto injection. Using laser, you will be able to deliver your gene of interest. And last